Okay, thank you very much, everyone, for your patience. And today it's our great pleasure to invite Alan Evans from Canada to give us a talk. Let me first just simply introduce his biology. And actually, we know each other in Abu Dhabi. We first know each other in Montreal, and he introduced some open neuroscience platform in Canada. And Alan Evans is a distinguished James McGill Professor at the Neurology and Psychiatry and Director Data Chair Neuroscience at the McGill University. He's the he's the scientific director of the Healthy Brain for Healthy Lives Institute, which he will introduce today, and also he's the scientific director of the Canadian and Open Neuroscience platform. I hope that our BrainMant project could have a good collaboration in the future with these two projects. He uses the high-performance computing to integrate the imaging, behavior, and genetics data in research, neurodegeneration, and neurodevelopment. Maybe you have already searched his Google Scholar. He has a very high H index, more than 200, and also he has like 2 million, more than 2 million citations. He's a fellow of the Royal Society of London, Royal Society of Canada, and the Cuba Academy Science, and also officer of the Order of the Canada. So let's welcome Alan Evan to give us a talk. Thank you very much, Evan.
Thank you very much for the very kind introduction, Sun Sensei. Good morning, everybody. Thank you for coming out at early morning in Japan. I'm going to give a talk today which will mostly focus on the infrastructural elements that were of interest. Sun Sensei expressed an interest in when we met in Abu Dhabi. I will show you some scientific applications that make use of this infrastructure. So the talk outline in general, I will talk about healthy brains, healthy lives. This is a large initiative at McGill. An overview, and then I'll give an example of the kinds of research that goes on. I'll talk about the infrastructure, and then I will talk about applications, national and international. And if I have time, I'll mention briefly the corporate spin-off that makes use of this infrastructure. So excuse the acronyms that I'm showing here, but there are a lot of elements that go into this, and it's important to set the scene so that you understand how they all interact with each other. So for this talk, the major elements are the platforms, the LORIS multi-modal databasing infrastructure, the CBRAIN high-performance computing platform, and I met with Sun Sensei when we talked about Neurolibre and evidence. That's open publishing. I actually won't talk about that today. Sitting on top of those platforms are many initiatives. Most important among those that I will talk about is the Healthy Brains, Healthy Lives initiative at the network level. And I will talk briefly about the Global Brain Consortium on top of all of that. On the right, you see many different initiatives that are making use of this infrastructure. I will mention some of those, but obviously not all of them. This figure will appear a couple of times in the talk because it's important to get the overall context of how these different components hang together. So starting off with Healthy Brains for Healthy Lives. This is a large initiative, $100 million initiative by the Canadian government. And its mission is to create a global center of excellence in neuroinformatics and to use that informatics infrastructure to translate discoveries in basic science to improve brain health. It's made up of 4 themes, a cross-cutting neuroinformatics and computational modeling theme, and then 3 application domains in neurodegeneration, brain plasticity, and population neuroscience, which includes psychiatry. As you can see from this slide, it supports something like 250 independent investigators and approximately 2,000 highly qualified personnel involved in HBHL. So it's a large project involving many different groups. Over the last 8 years, it's been in existence. It's published something like 2,500 peer-reviewed publications have come from HBHL. The fundamental perspective of HBHL is that because of the phenomenon of pleiotropy, multiple genes can be associated with any particular disorder and brain state, which makes life very difficult to interpret. In fact, genes typically code for a mechanism or a pathway or an enzyme. And those pathways are the ones which are found expressed in the different brain states. So it makes more sense to deconstruct our investigation into brain processes from a mechanistic level rather than a disorder-based level. So just a few of the people involved in the theme one are involved in network modeling, multi-modal network modeling of pathology propagation. One of them, Yasser Ituria Medina on the top left. I will use one portion of his work to illustrate the general principles, but there are many other different scientists involved in HBHL, which is what we call healthy brains, healthy lives. The theme two is the mechanistic models. As I said, I will talk about some of the work that's going on from Yasser Ituria Medina. That work started its origins from the recognition from the work of Randy Buckner in 2009 that on the left, areas with high functional connectivity are also areas which on the right show high beta amyloid deposition. So the implication there is that they have networks and hubs and nodes in those networks are places where there's a lot of activity, functional activity, and that gives rise to the greater deposition of misfolded proteins. So Yasser developed an epidemic spreading model of beta amyloid propagation, which made use of a large number of PET images of beta amyloid deposition. The brain regions segmented by a typical canonical brain atlas for each subject. The networks are the typical diffusion-based network connectivity, and the modeling framework essentially did a time-lapse analysis across all subjects. So you convert a cross-sectional study into a longitudinal study and use this basic model to model the propagation of the beta amyloid from node to node within a brain network. Here's the epidemic spreading model itself, and it essentially accounts for the differential equations to account for the propagation from node to node throughout the network. It has two important parameters for each network connection, the A-beta production rate and the A-beta clearance rate from any given node. And this is the important conclusion from that analysis of a large population of data from the ADNI data set. And you look at the top left first. The point here is that across four categories, clinical categories, from healthy controls on the right, early MCI, late MCI, and then full Alzheimer's disease, there's not much difference in the production parameter of beta amyloid in that region. On the other hand, the top right, the clearance graph shows a steady change in the clearance rate of the beta amyloid from healthy controls, reducing down to full-on Alzheimer's disease. So the important point about this is that the buildup of beta amyloid in any given region is not a function of overproduction. It is a function of underclearance, and that has significant ramifications for drug design. On the bottom here, you see expected results, given what I just described, that the production rate for different levels of ApoE4 status, 0, 1, or 2 copies, doesn't change much. But the clearance rate for those 3 categories is dramatically different for people with no ApoE4 down to people who have 2 copies. So this would be what you'd expect to see if clearance was the critical determinant of the buildup of beta amyloid. What this led us to further look and investigate this phenomenon was that clearance from the glymphatic system and from the vascular support system, that is a critical early determinant of the buildup of beta amyloid. And the results from that study are shown on the right here from Ituri Medina in 2016. And it is to be contrasted with the well-known JACC graph from 2010 and 2013, which look at the stage of events within the Alzheimer's disease pathophysiological evolution. And you see that in the graph on the left, blood flow wasn't considered to be a critical determinant. Our results on the right show that it is in fact the earliest determinant of beta amyloid buildup. Just to take the story a little bit further and more up to date, as we know now, that there are 2 particular misfolded proteins involved in the pathological process of Alzheimer's disease, amyloid and tau. And we now think that tau is perhaps more directly associated with the pathophysiological process than amyloid. It comes earlier in the process to years prior to symptom onset. It's more focally distributed. It has a stronger association with brain health, and it tracks well with cognitive status. So you can do the same sort of thing that I just described with beta amyloid with tau protein. So in subsequent studies, Jake Vogel, the PhD student that I just showed, used the SUSTAIN analysis environment developed by Alexandra Young. And this essentially allows us to take a large amount of cross-sectional data in Alzheimer's disease and break it down into subtype and stage modeling across the cohort. And what Jake showed using this approach was that we could identify 4 subtypes of Alzheimer's disease with different propagation trajectories. Quite a few publications arose from Jake's work. And in essence, it showed you that within the epidemic spreading model, if you use a best epicenter for the propagation for each of those 4 subtypes, you get a better fit to the data than if you assume one single entorhinal epicenter, which is what we might have thought at the outset of this process. So there are 4 subtypes which can be dealt with independently. That's the end of that one example of the kinds of research that goes on within HBHL, Healthy Brains, Healthy Lives. In fact, as I said, there are 240 PIs, principal investigators, involved in HBHL. So, of course, you cannot cover the entirety of all the work that's going on. That's just one example of how we're approaching the pathological clinical research.
So I have a question. Is it okay?
Yes, please.
So in your continuous research, so you are also making a model, and I'm very interested in the future work. So that means that based on the diffusion MRI and the connectome, and so you have the PET data, and I seem to be mostly the basic model is finished. But what is a more advanced topic or the more interesting topic for your viewpoint?
Right now, Yasser Ituri Medina in particular is incorporating information from the omics domain, from transcriptomics, metabolomics. All of those factors can be brought into the epidemic spreading model. And he can also incorporate data from postmortem data. There's a wealth of publications that have updated this work that you can check if you look under Ituria Medina.
Yeah, that is interesting. And in the Japanese people is investigating the new tau probe for the PET data. So how do you think this kind of development of the more accurate tau proteins propagation observation from by using a PET?
Well, I mean, it obviously depends on the quality of the tracer, how specific it is to different pathological moieties. And there will be a continuing evolution of new tracers and more tracers. So the basic data acquisition will continue to improve on the one hand. I think where we are going on the analytics side is what you have here right now is a parametric modeling framework. People will start increasingly to use potentially non-parametric AI type approaches. As I'm sure you're well aware, the issue then is how do you interpret the non-parametric approach and assign results to particular deficits in particular biological, physiological components. If we can move to generative AI with parametric models in there, then I think we can take advantage of the strength and the power of AI while retaining a parametric perspective. I think it's absolutely essential that you have to have a biological interpretation pre-hoc, not post-hoc. Trying to retrofit the biology into an AI model when you haven't set up a prior architecture for the parameterization of the data, I think is a dangerous way to go. Anyway, that's for, I guess, a broader discussion. I'm going to continue with the discussion of the informatics that underpin this science.
Thank you very much.
So that was just one example from about 240 principal investigators. To support that community, you need significant information technology, IT infrastructure. And what we've developed for HBHL, it preceded HBHL, but it has been advanced over the last 8 years through HBHL, is what we call NeuroHub. And there are many components to it. I will focus specifically on two of these here, LORIS and CBRAIN. What are they? LORIS is a multimodal database management environment that has been in continuous evolution for the last 20 years or so. It's continuously evolving. And what you see, it allows, on the left, it allows us to capture multimodal imaging data, summary genetic epigenetic data. I mean by that SNPs and CNVs. We do not store all of the raw genomic data. We don't want to. We only want the derived SNPs and CNVs, that kind of information that will be compared with the phenomic information, the imaging and the behavior and so on, within the context of an HBHL type study. Carrying on. Imaging, genetics, epigenetic information, behavioral clinical information. There are about 1,000 behavioral instruments incorporated into LORIS right now, and more can be added by new users. Biospecimen information, I'll give you an example of that, of how this is used to support a tissue biorepository later on in the talk, and electrophysiology, specifically EEG. So LORIS is a multimodal databasing environment that is coupled with CBRAIN. CBRAIN is a portal to high performance computing and now cloud services. So you can submit large scale jobs on a compute cluster infrastructure. I will talk a little bit more about that in coming slides. As an example of how this infrastructure is used to support HBHL, McGill was the first site to request all of the UK Biobank data. Prior to our request, they were providing UK Biobank data that was specific to a particular hypothesis proposed by a potential user of the UK Biobank. In our case, we had 240 PIs. We need all of the data. We will then be responsible for the dissemination of subsets of that data to our individual PIs. And I think the UK Biobank at the time, this is about four years ago now, realized that it was so popular to distribute the UK Biobank data that they were having an issue of how to accommodate many, many users in many, many sites. And it was the hypothesis-driven approach wouldn't scale, whereas the approach that we were using, send us all the data and we will take it from there for our internal group, was a better way to go. So we have all of the UK Biobank data that we service to the HBHL community. CBRAIN, as I said, is a portal to the high-performance computing infrastructure, initially of Canada, but now it's expanded and it has nodes in Europe and the US in particular. We would be very happy to add Juntendo as a node in CBRAIN. It would make, I think, a great advantage for lots of people to use Juntendo HPC resources. Essentially, CBRAIN allows data to be anywhere, computing to be anywhere, and users to be anywhere. I'll give an example of that in a moment. And as I said, now it's not just HPC, it's also cloud services. The data are managed by a boutiques environment, which is a containerization environment, which allows one to take complex workflows, invest those into a containerized environment, and using the boutiques platform that was developed at McGill. And at this point, this is a little bit out of date, this slide, but you see that the number of tools available at about 160 different work packages. Those started life mostly as analysis of fMRI or tractography or PET data imaging. But increasingly on the top right, you see that we're adding genomics and transcriptomics workflows, RNA-seq type of analytic engines. And there's a little bit more detail on some of those engines. I mentioned RNA-seq, and then there's scRNA box and methylation pipelines and Cell Ranger. These are all tools that the omics community want to use. You can find out all of the details on this by going to the CBRAIN website. At this point, the CBRAIN is established and has over 2,000 users around the world. 193 sites in 59 countries are making use of CBRAIN resources. And last year we added DataLad, the Stanford data repository has a direct link between DataLad and CBRAIN, such that if the data is in DataLad now, it is also accessible through CBRAIN and can be processed through CBRAIN. So that was a major step forward for us last year. So we just added last year eBrains, and I'm sure you're familiar with the eBrains infrastructure developed by people working with my close colleague, Katrin Ammons. I will talk more about Katrin and her community a little later in this presentation. So people can get access to large data sets through these different front doors, and CBRAIN can analyze all of those data from those different sources. Just to make the point here, the data at this point can be resident anywhere in the world, and the compute can be anywhere in the world. So in the use case at the bottom, I've put in the data can be in brain mines in Japan. The computing can be performed elsewhere. The data can be transferred in a volatile manner temporarily to the HPC or compute resource, but it is not retained there. As we've increasingly reached the point where you want data is too big to transfer permanently. There are national and regulatory concerns about data being transferred outside of national boundaries. So these are not retained, they're just processed. And the results are then made available back to the user. An example of one particular tool, I'll be talking about this a little bit later in the talk. The example here is what we call HIP-Unfold. HIP-Unfold is an analysis tool developed in the lab by Jordan de Cracker. This is a tool for segmenting the hippocampus into its subfields on essentially a flat map modeling environment. That's for people who are interested in hippocampus. I can talk more about that if questions arise. But the important point from this slide, from an informatics perspective, is that the data can be, in Japan, computing can be performed on any CBRAIN node in the world, and results are restored back to the user in brain minds. CBRAIN has been around a long time with a growing user base. This paper is from 2014 that describes CBRAIN. There are many more papers that are available that people can go to. I'll send these slides out for anybody who wants to follow up in more detail. Both CBRAIN and Loris are open source tools available through GitHub. I'll now move on to some applications describing some initiatives that make use of this infrastructure that I've just described. Some of them I will talk about here. I talked about healthy brains for healthy lives. I will talk about the CBIG top left, and I will talk about the Canadian Open Neuroscience platform. But time doesn't allow me to go into great detail on some of these other ones. The Canadian Consortium of Neurodegeneration and Aging, the bottom left, that's an initiative of about 450 dementia researchers across Canada who are making use of this infrastructure. So to get a little bit more detail on some of these initiatives, the CBIG initiative is the Clinical Biological Imaging and Genetic Biorepository, CBIG. And at this point, it has 200 cell lines, 6,000 donors, 125,000 tissue samples that are made publicly available in an open capacity. They have 130 collaborations across the academic and industrial sectors who are making use of this open tissue biorepository. All of this is supported specifically by the Loris database and to a lesser extent, the CBRAIN analytic engine. Most people just want to get access to the tissue itself as a biorepository, so they're more interested in the data than downstream analytics. Nevertheless, that is available to them if they wish. One of the initiatives that is still relatively early in gestation is the so-called CANOE initiative, where we're combining the Pan-Canadian Genome Library, which is obviously focused on genomics, and the current CBIG model, which is mostly focused on phenomics, imaging and behavior. And obviously, where we want this to go is to have one ring to bind them all, one platform which supports both the genomic and the phenomic data. We're using this in one particular context right now, that of rare diseases. I have to stress that rare diseases are rare in the sense that not many people have a rare disease. However, there are over 7,000 rare diseases. So what you want to do in this context is to allow oneself to assemble data from multiple sites to build up the statistical power of studying any one rare disease. But make no mistake, there are 7,000 rare diseases. So getting this in place at a national level is an important initiative to deal with a large number of conditions. On the bottom right, I don't have time to go into great detail, but a lot of this infrastructure that I've talked about is focused on brain. There is very little in it that is brain specific. And we're using the same infrastructure to support a heart initiative in Ottawa called the Brain Heart Interconnectome, and that is making use of all of this infrastructure to support heart and more recently oncology applications. This is relatively early, but it does demonstrate the data agnostic nature of this infrastructure, that it can be used in heart applications pretty much out of the box. The Canadian Open Neuroscience platform has been around for about 10 years. And this was predicated on the notion that we want to share data openly with people around the world. And this required us to develop the organizational, regulatory documentation associated with open data sharing of data, be it imaging, behavior, or genetic information. And the CONP has been doing that for about 10 years now. It also is the birthplace of the NeuroLibre evidence open publishing concept that Sun Sensei and I talked about in Abu Dhabi recently. All of that has arisen directly from the Canadian Open Neuroscience platform. And this, of course, involves training and international partnerships, as well as data and tool sharing. The tools that are shared, essentially the ones that I just described, which are on CBRAIN, and about 100 different data sets which are available to the neuroscience community in Canada. There are numerous publications on the Canadian Open Neuroscience platform. Some of them are shown here, and you can find these from the slides when I send those slides to you for a further analysis. The last one that I'll briefly mention is EEGNet. This is an open data platform for analyzing EEG data across individual subjects. EEGNet is built on top of the Canadian Open Neuroscience platform. So it's very much focused on EEG data, but it's using the same tools that we've used for behavior and imaging in CONP. On the international scene, there are a number of projects worthy of mention. I mentioned already our use of the UK Biobank within Healthy Brains, Healthy Lives. But this infrastructure is also used to support some larger initiatives, like the infant brain imaging study of autism in the US, the Adolescent Brain Cognitive Development Initiative in the US, the Healthy Brains and Child Development Study in the US. The last one there, the Healthy Brains Child Development Study, is relatively new. It's about 3 years old now. They're still in the process of data collection. This is making use of the LORIS and CBRAIN infrastructure in particular, as well as the commercial version of this for data dissemination. I'll talk about that at the end of this talk. The one that I will now briefly talk about HIBALL here and the Global Brain Consortium here. These are two other initiatives that are making use of this infrastructure. HIBALL started life about 2013, so that's 12 years ago. We published this in Science. This is the big brain. This was at the time an ultra-high resolution 3D human brain model. It's essentially 20 micron histology data in 3 dimensions, isotropic resolution in 20 microns in each of 3 dimensions. At the time when this was first published, this was a terabyte in size, and that was a big data set. Nowadays, a terabyte is not so big, and people are creating larger and larger data sets. But as far as I know, there's still not yet a higher resolution 3D data set of the whole brain in existence. And HIBALL is the big brain has been around now for publicly since 2013. The work that went into that was another 10 years prior to the publication of this paper. So HIBALL is an outgrowth of the big brain collaboration with Katrin Amundsen. Katrin Amundsen, some of you will, I'm sure, know that she was the scientific director of the European Human Brain Project that has recently come to a logical close. And Catherine and I have been working together on HIBALL related projects for about 25 years. The HIBALL is essentially an expansion of Big Brain to include other types of data, multimodal data acquisition. What you're looking at here is the Big Brain in 3D. It's showing the hippocampal subfields that were derived by Jordan de Cracker. I talked about his HipUnfold pipeline earlier. And what you see on the bottom left is the hippocampal, on the bottom right, sorry, the hippocampal subfields. And you'll notice something that surprised me when this was first produced, that the hippocampus has sulci. It has folding patterns similar to the cortical surface, which I was not aware of until this high resolution 20 micron map was produced. The HIBALL network has about 900 researchers in 45 countries who make use of data that's acquired within the HIBALL initiative. An example of this kind of work is the cortical layer analysis, where we used a deep learning approach. Konrad Wagstill in my lab at the time and Hannah Spitzer in Katherine's lab at the time worked together using a one-dimensional convolutional neural network to identify individual cortical layers in the Big Brain data set. What you see here are the six layers of the human cortex identified using a CNN AI convolutional neural network approach, and you can see the layers shown there. What this shows is a validation and a confirmation of the results that were obtained by that AI approach, where we looked at the comparison with traditional histological cortical layer analysis from von Economo, and the same from analysis of the Human Connectome project using MRI. And what you see here, the two graphs show you a good concordance between Big Brain cortical thickness using AI, fully automated analysis, and either von Economo or HCP. On the hippocampus I've just shown you before, this is the hippocampus of the Big Brain. And this is the work that was done by Jordan de Cracker, postdoctoral fellow, who showed the hippocampal segmentation and the unfolding of the hippocampus into a surface analysis framework that is being widely adopted. Just a brief mention of one of the other projects that is going on within the context of HIBALL. This is work by Justine Hansen, working with her supervisor, Bratislav Mizik. This was the use of PET to identify and map 19 receptors and 9 neurotransmitter systems from PET data acquired from 1,200 different PET maps from multiple sites, some in Japan. So in summary, the HIBALL is providing maps and models to the community, some of which I've just described, whether it be histology or cortical layers or three-dimensional chemoarchitectural maps. Those are all available through HIBALL. And it also provides various analytic tools, some of which I've just described for you. You can find out about all of these from the HIBALL human brain mapping website. We had at this point 9 workshops, annual workshops on HIBALL. The last one we just had was in Berlin, in Germany. The next one that's coming up is in Montreal again. So by all means, if you're interested, please do join us in October in Montreal to see more of this. The Big Brain project, the HIBALL project, I should say, has produced at this point about 130 publications since 2020, some of which are illustrated here. And we're increasingly concerned with globalizing brain research using these tools. Katrin wrote a review on this. I wrote the section on globalization capabilities, and a lot of the infrastructure that I've just been described will be central to the globalization of these initiatives. One of the things that I think it's worth mentioning, I've already alluded to it, but a little bit more specifically, HIBALL was the environment in which we developed the internationalization of the CBRAIN platform to a higher degree. This came because on the left-hand side in North America, we have, as I've described, LORIS, the CONP platform, the CBRAIN. Katrin's European partners have equivalent infrastructures with eBrains and the Phoenix supercomputing portal and so on. And obviously what we wanted to do was to make these compatible. Well, at the outset, about 4 years ago now, when we started this project, data on either side of the Atlantic were non-harmonized, different user authentication and authorization, costly transfers or disallowing copying of data, non-interoperable HPC systems. So there was a lot of incompatibility. So we could wave at each other, but we could not share data transparently. Over the last 4 years, we've been working to harmonize this process. And now we do have harmonized authorization, transfer protocols, interoperable HPC systems. All of this has given rise to what I described earlier, where data can be essentially anywhere and analyzed from anywhere using the CBRAIN portal. I won't waste your time going into the weeds here. Did I show this? Yeah, I already showed this. This was the task can be anywhere, data can be anywhere, and processing can be anywhere. So I'm coming back to the overarching schematic, which allows you to put all of these things in context. One of the things that I have not mentioned yet is at the top there, the Global Brain Consortium. So I'll finish up with a few thoughts on what the Global Brain Consortium is and where it's going. The Global Brain Consortium, it's an organization for democratization of brain mapping practices into low and middle income countries, LMICs. It's focused right now on EEG because EEG is inexpensive and has high temporal resolution. In due course, we will want to expand that to incorporate at least MRI imaging. But for now, it's focused on EEG. And it wants to provide increased access for LMICs to electrophysiology data, tools, methods, and infrastructure to foster collaborative neuroscience internationally. Want to promote best practices for international open science and data sharing and applications in disease, disorders, and discovery. The co-directors are Pedro Valdez Sosa, a world authority on electrophysiology, and who has a lab in Cuba and in China. Myself, Katrin Ammons, the number of people that I've already mentioned in passing. The projects that we're focused upon mostly are global EEG norms, COVID-induced brain dysfunction, data-driven electrophysiological approaches for individually tailored diagnosis by Paolo Rossini and Yasser Turia Medina, and some other ones. You can see them all here. Some examples here. Born in Bradford, this is a study of over 13,000 subjects from economically deprived areas of Bradford, capturing all of their genetic, cognitive, and school healthcare records. And we're building on this platform to collect EEG data in schools across that cohort. On a more global scale, an agreement was reached in 2022 between the IFCN and the Global Brain Consortium to foster the international dissemination and harmonization of EEG data acquisition. This is a continuing problem in EEG, that the data acquisition methodologies are incompatible in different countries. And a significant amount of work has been done by the GBC to harmonize across these different regions to standardize data acquisition and therefore data analysis. I will finish with just a brief mention of LASSO. LASSO is a commercial spin-off from my lab that was set up to allow for the broader dissemination of these infrastructures, and particularly into areas where scientifically it might not be particularly interesting to brain people. But if you're interested in heart or oncology, then LASSO would be able to provide those same tools on a commercial basis. And one of the important points to make is that LASSO was chosen by the National Institutes of Health in the US to provide the data dissemination infrastructure for ABCD and HBCD, which I've mentioned before. These are developmental cohorts, normal developing cohorts, and cohorts which look at the influence of opiate addiction on child brain development. LARIS is involved in the internal management of data. LASSO provides the infrastructure to support the dissemination of the data collected in these large initiatives to between 2,000 and 5,000 potential users of HBCD and ABCD data. So I know I've covered a lot of territory in this overview, and I think this slide, again, I keep on showing it, but it does allow us to keep track of what they all mean and how they all hang together. So I thank you for your time. I'm more than willing to talk about potential collaborative opportunities between our infrastructure and brain minds. Thank you very much for your time.